Hello, and thanks for joining us today for the latest Technology Networks webinar, exploring tumour innovation with tissue clearing and 3D imaging. I'm Dr. Kate Harrison, science writer for Technology Networks, and I'm here to moderate the event. I'm very pleased to have Dr. Sophie Chauvet joining us today as your presenter. A warm welcome to you. Dr. Chauvet completed her doctoral studies in developmental biology at Aix-Marseille University. Her research is predominantly centered around investigating the mechanisms and molecules involved in axon growth and guidance. Currently, her research endeavors are directed towards comprehending the intricate interplay between the nervous system and solid tumors, particularly in pancreatic cancer. This groundbreaking research in the field of cancer neuroscience holds promising prospects for future investigations and holds the potential for substantial clinical implications. Following the webinar, we will have a Q&A session and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the right-hand side of your screen and click Send, and we will answer as many as possible during the time available. For any questions we don't have time to get to, we will make sure you are contacted offline with an answer. If you experience any technical issues, please click the chat icon in the bottom right corner to request support. Please remember, you can ask questions at any time, even while watching the webinar on demand. So without further ado, I will now hand over to Sophie.
Hello everyone, I'm delighted to have the opportunity to share our work in this webinar series organized by Miltenyi Biotec. Today, I will show you how tissue clearing and light sheet microscopy helped us to better understand the importance of innervation in tumor progression. In the team, we focused to understand the importance of the peripheral nervous system within solid tumor. What is the peripheral nervous system? The peripheral nervous system plays a vital role in facilitating communication between the central nervous system, the brain, the spinal cord, and the other area of the body. It serves as a connection between the central nervous system and organs, muscle, and skin. This system is comprised of two main divisions, the somatic nervous system and the autonomic nervous system. The somatic nervous system is responsible for transmitting sensory and motor information to and from the central nervous system. The sensory system allows us to perceive pain, and it will allow us to protect us against harmful stimuli as fire or hot water. On the other hand, the autonomic nervous system regulates involuntary body function, including heartbeat and breathing. It operates autonomously, allowing us to breathe without thinking. The peripheral nervous system has such important function in physiological conditions that it was tempting to look at its function in tumors. A little bit of history. Back to 1897, using a technique called methyl blue staining, it has been demonstrated the presence of nerve fibers within tumor, as you could see here. Few studies confirmed this at the beginning of the 20th century, especially this one in 1930. They confirmed the existence of tumor innervation and hypothesized that these fibers might represent newly growing nerves, without properly showing it. However, it was until the beginning of the 21st century that the field of cancer neuroscience truly began. Indeed, electron microscopy enabled researchers to provide concrete evidence of tumor innervation, as you could see here. A breakthrough study published in 2013 by the team of the regretted Paul Frenette demonstrated the innervation of prostate tumor by the peripheral nervous system, and it also showed the impact of tumor initiation and progression. 1 year later, similar findings were reported in gastric cancer. Since then, the cancer neuroscience field is flourishing with an impressive amount of amazing publication. Here, you could see several techniques that have been used to visualize tumor innervation. it is important to have in mind the scale that we will consider to look at the neuronal compartment within tumor. Here, you could see the scale of the nerves entering into the tissue. Then, nerves will divide into axon bundles, that you could see here, and individual fibers or axons. The individual fibers secrete at their extremities the neurotransmitters which exert their effect on the target cells. Several mechanisms of tumor innervation has been proposed. The data that I am showing you on this slide has been done in human pancreatic tumor. In the first analysis, they showed that there is the same amount of nerves in healthy and tumoral tissue, leading to think that tumors invade the neural region. In another studies, they showed an increase of nerves in tumor region compared to healthy region, leading to think that nerves are able to regrow within tumor. But the images I showed you only provide a two-dimensional view and do not consider the three-dimensional network of the peripheral nervous system, which make it difficult to look at the remodeling of the neural compartment. So, we decided to develop 3D analysis to look in a different way at neuronal network in tumor. To do so, we focused on pancreatic ductal adenocarcinoma, PDAC, which represent 85% of the pancreatic tumors. In this type of cancer, it is crucial to explore novel therapeutics approaches, considering that it has been projected to become the second leading cause of cancer-related deaths by 2030. We employed a genetically engineered mouse model known as KIK model, an overexpression of the mutated form of the oncogene KRAS in pancreatic cells induce a process called transdifferentiation, whereby acinar cells transform into ductal cell-like cells, leading to the formation of metaplasia called ADM. Subsequently, this metaplasia progresses to neoplasia known as panine. The deletion of tumor suppressor genes INC4A-ARF further promotes the development of tumor, the PDAC by itself. The KIK model successfully recapitulates the stepwise progression observed in human pancreatic cancer. On these pictures, you can observe a mouse wild-type pancreas with the spleen, which serves as a reference point for tissue orientation. The spleen aided in identifying the head and tail region of the pancreas. This distinction is crucial as the head region houses a network of nerves that enter into the pancreas. Here is a cleared pancreas, allowing better visualization analysis in 3D. In order to visualize and analyze the stages of disease progression, we combine immunostaining, tissue clearing by using ID-Scope+ protocol. Then we image the tissue in 3D by light sheet fluorescent microscopy by using, of course, the UltraMicroscope blaze of Miltenyi Biotec. We use antibodies against cytokeratin-19 and alpha-smooth muscle actin, alpha-SMA, with topo 3 nuclear contrast staining. In the healthy region of the pancreas, cytokeratin-19 stains the epithelial cells of pancreatic ducts. You could clearly see the network of the duct in green, and alpha-SMA is expressed by the vessels in red. In the different region of the KIK pancreas, we observed a distinct pattern. Cytokeratin-19 expression was found in cancer cells, and alpha-SMA was expressed by cancer-associated fibroblasts, the CAFs. On the optical section, here, you could clearly see that the intrinsic fluorescence of the tissue could serve as a reliable marker for recognizing and distinguishing different stages of the disease, the PDAC there. We used the image of the autofluorescence of the tissue to develop deep learning techniques for the automated segmentation of tumors within tissue samples. Our primary objective was to achieve full automation in this process. We started by pre-processing the raw image stack obtained from autofluorescence staining. This pre-processing step was crucial in enabling efficient analysis of the entire dataset. We then employed the LabKit program, which allows us to segment the entire tissue slice by slice. In our system, LabKit program was not efficient to segment the tumor part. So, to identify tumor nodules within each slice, we developed a specialized UNet model. By using Imaris software, we merged the result obtained from the two methods and reconstruct the 3D tissue. This enabled us to identify and locate tumor nodules in red within the entire pancreas in white. We then focus on the innervation of the pancreas. In my talk today, I will focus on one part of the autonomic peripheral nervous system, the sympathetic nervous system, which regulates the flight or fight response. The sympathetic system responds to environmental stress, which are known to play a role in tumor occurrence. We employ tyrosine hydroxylase antibody staining. TH is an enzyme involved in biosynthesis of norepinephrine, one of the main neurotransmitters of the sympathetic nervous system. On this picture, you could see that TH is expressed by all postganglionic sympathetic neurons of celiac superior mesenteric ganglion complex that supply the pancreas. Nerve bundles enter into the pancreas by following blood vessels, which are in red on this picture. Quantification through equidistant optical section to cover the entire pancreas showed a striking heterogeneous distribution of nerves with a higher density of nerve innervation at the head of the organ, which gradually decreased towards the tail of the pancreas. We then looked at nerve network in kick pancreas. Even if the overall pattern of nerve in kick pancreas appears disrupted compared to wild-type ones, probably because of the perineural invasion, the main shape of the network was conserved. Indeed, we could recognize some pre-executive nerve patterns at the one enumerated here, and you could clearly see the ones innervating the spleen which travel through the pancreas in wild-type and in kick. As we showed in wild-type condition that the repartition of nerve innervation is heterogeneous, we reconstruct the total volume occupied by the nerves. The whole volume didn't vary between control and kick pancreas, as you could see on this graph. To see how this data could fit with the 2D data that I showed you previously, we quantified the quantity of nerve outside and inside the tumor. Here is one example in which you could see the reconstruction of tumoral lesion in transparent green, the nerve outside the tumor in red, and the intratumoral nerves in yellow. There is a huge heterogeneity in nerve density between the diverse tumoral nodules of the same pancreas. Here, almost not, and here, a lot. There is also a huge heterogeneity between the percentage of nerve inside the tumor and outside, depending on the analyzed pancreas, as you could see on this graph. To summarize, the total volume of the nerve stays almost the same, but the quantity of nerve engulfs the tumors vary a lot. This result suggests that the intratumoral sympathetic nerve bundles frequently reported in PDAC may correspond to pre-existing nerve bundles become embedded into tumor. We then focus on terminal fibers by looking at higher magnification at the level of pancreatic lobule. We can appreciate the very dense meshwork of sympathetic fibers in wild-type pancreatic lobule. The picture in a pancreatic kick lobule shows the same kind of network, where we found local hotspots of high sympathetic innervation that are never seen in the controls. These hotspots corresponds to axon innervating a neoplasic lesions as shown by the autofluorescent signature of the tissue and confirmed by cytokeratin 19 staining. The 3D reconstruction of the neuronal network in the lesion showed an hyperinnervation. Thanks to Imaris software, we are able to systematically reconstruct the sympathetic axon network in red, as well the vascular network in green, and the contact surface between axons and vessels in yellow. From this reconstruction, we could extract a dozen of parameters that describe the architecture of neuronal network and proximity interaction with blood vessels. We performed this reconstruction in the asymptomatic region and in distinct lesions of the kick pancreas, including metaplasia, neoplasia, and PDAC region that I mentioned before. The blue staining here corresponds to the empty space found in the different lesions. If we focused on the neuronal network, we see an hyperinnervation of metaplasia and neoplasia, as we've shown before. On the other hand, fewer sympathetic fibers are found inside PDAC nodules. Z-score analysis shows several differences between the analyzed region. As example, there is an increased density of axons with more branching, indicating hyperinnervation of metaplasia and neoplasia lesion. A hierarchical clustering on this data revealed three groups with different innervation patterns that coincide with healthy in blue, pretumoral lesion in yellow, and tumoral lesion in red. To make a long story short, we showed that this axonal remodeling didn't occur by an increase in neurogenesis, but by local sprouting of the pre-existing axons. Next, we ask how the sympathetic nervous system influences PDAC progression. We selectively ablated sympathetic nerve fibers in kick mice. In sympathectomized mice, there is an increase of tumor weight. Moreover, sympathectomy reduced survival of tumor-bearing mice, blue curves, compared to the black one. And also, kick mice is a non-metastatic model. Sympathectomized mice show the formation of metastasis in distant organs, including the liver and the peritoneal cavity. Thus, the sympathetic nervous system exerts a protective function against PDAC development, pointing to an anti-tumoral effect of the sympathetic nervous system. One hypothesis to explain this result is that the norepinephrine released by sympathetic fibers could act directly on cancer cells or indirectly by playing on tumoral microenvironment. We showed in vitro that norepinephrine didn't play so much on tumor cells, so we focused on the tumoral microenvironment. Given the potential function of neural signals in modulating immunity, we tested whether the response to sympathectomy involved the immune system. We focused on macrophages and especially on CD163 positive tumor-associated macrophages, which are known to be pro-tumorigenic. We have once again performed immunofluorescence tissue clearing and 3D analysis by using F480 as a pan-macrophage marker and CD163. we showed a significant increase in CD163 positive macrophages in PDAC region, as you could see here in red. We showed in vitro that norepinephrine downregulates CD163 expression, meaning that norepinephrine blocks the transition of the inflammatory status of the macrophages to the anti-inflammatory protumoral function. Taken together, these results indicate an immunomodulatory function of norepinephrine released by sympathetic nerve terminals that will contribute to limit the protumorogenic and immunosuppressive CD163 positive macrophages in pancreatic pre-neoplastic tissue. To test whether the increase in CD163 positive macrophage contribute to the worst outcome of sympathetomized mice, we used liposome vectorized with an anti-CD163 antibody to selectively deliver the chemotaxic agents doxorubicin, DXR, lipo, to CD163 positive macrophages. On this graph, the blue line corresponds to denervated kick mice treated with a control liposome, and like I explained earlier, we can see a decreased survival of the denervated kick mice compared to non-denervated ones. Black line. Then, if we treated kick mice with doxorubicin, blue dot line, we found that the selective depletion of the CD163 macrophage population was able to restore the normal survival of the denervated kick mice. On the basis of this data, we propose a model in which KRAS mutation induces transdifferentiation of the acinar cells, which increase inflammation. Indeed, at that step, the macrophages have an inflammatory status and an anti-tumoral effect. At metaplastic and neoplasic stages, there is a remodeling of the sympathetic fibers which could secrete norepinephrine. Norepinephrine will inhibit CD163 expression. It inhibits the switch of the macrophages towards an immunosuppressive effect, explaining the anti-tumoral activity of the sympathetic fibers. In PDAC, there is less sympathetic fibers remodeling, allowing the presence of CD163 positive macrophages. And it has been shown that these macrophages can suppress T cells mediated anti-tumoral immunity by activating the immune checkpoint CTLA4. To conclude, sympathetic axons have the ability to sprout and innervate metaplastic and neoplastic region in the aim to slow down the progression of PDAC by controlling the local immune environment. To finish, I would like to thank all the members of the team, especially people who has done the work, all our collaborators, and you for your attention.
Thank you very much, Sophie. I'm sure that everyone will agree that there were some excellent points raised during the presentation. So we will now begin the Q&A session. To submit a question to our speakers, you should enter your question into the box on the right-hand side of your screen and click Send. We will answer as many questions as possible during the time available. So we have actually already had a few questions submitted, so I'm going to move straight onto these. But if you still have a question you'd like to submit or you think of one, you can still submit questions. Okay, so we've got this one that says, thank you for the informative presentation. In relation to the IDISCO clearing method you discussed, are there alternative clearing methods that can be used for clearing the Oh, dear, that's disappeared. Sorry, that question's disappeared. Well, I'll move on to a different one while we get that one back. Sorry about that. Okay, so a different question. Based on your study, what potential therapeutic strategies could be explored to modulate sympathetic innervation and its protective response against tumorigenesis in the pancreas?
Thanks a lot for these questions. So it's a little bit difficult because as the sympathetic nervous system has an anti-tumoral activity, to get a response against tumorigenesis, the best will be to activate the sympathetic nervous system by using agonists as isoproterenol as example. But it's not so simple because as I explained you during my talk, the sympathetic nervous system mainly at the level of the metaplasic and neoplasic lesion, so at the beginning of the disease. And in fact, in patient, almost when the PDAC is detected, it is also at the tumor stages for the patient. So once the sympathetic nervous system does not innervate or innervate less the tissue. So what we could imagine, in fact, is more to try to understand better how the sympathetic nervous system could remodel. And the best things would be to induce the remodeling of the sympathetic nervous system at tumoral stages. But this for sure will be not so easy. So use it as a therapeutic target is not maybe the best for the PDAC.
Okay, fantastic. Thank you. I've solved the little technical issue we had, and the question that I was reading out before I've managed to get back. So to recap that one, in relation to the IDISCO clearing method you discussed, are there alternative clearing methods that can be used for clearing pancreas or similar organs? And what is the typical duration of the clearing process?
So when we started, we used different kind of techniques. So we've tested the cubic techniques and the IDISCO plus techniques. And for sure, the results that we got was better with IDISCO plus. So we keep to continue by using these techniques. And in fact, it seems that methanol treatment and hydrogen peroxide treatments that are performed during these techniques are really important to get a nice antibody staining. And for the clearing, so we tested two different components to perform the clearing. One is called DBE for dibenzyl ether, and that's the one that is the best to get the clearing, and it takes around 24 hours to 1 day to 2 days, depending. And we also test another one, which is known to be less toxic than the DBE, which is called ECI, for ethyl-3 prop2NO8. So sorry for this really bad name. But with this one, the clearing we got is less nicer. So we prefer to use DBE. And now we are trying new techniques which follow, which is based on the IDISCO plus protocol, but with avoiding methanol and paraformaldehyde treatment in order to try to keep endogenous fluorescence with the transgenic line strain with which we work with. And for the moment, we got nice result with this, but this allows us to get the autofluorescence of the tissue, but it's less good when we want to test with antibody staining. So ongoing work, I would say.
Okay, brilliant. Thank you very much. Next question. Have you tested whether similar mechanisms of sympathetic innervation exist in other types of cancers, or is it specific to pancreatic ductal adenocarcinomas?
So we haven't tested ourselves this, but for sure it has been tested by several other teams, as it has been well demonstrated that sympathetic nervous system may innervate other kind of cancer as breast cancer, gastric cancer, and also prostatic cancer. And interestingly, in this kind of tumor, they have observed the opposite effect. So in our case, the sympathetic nervous system has an anti-tumoral activity, but in this other kind of cancer, it has a pro-tumoral activity, meaning that in this other kind of cancer, it could be interesting to block the activity of the sympathetic nervous system more than to activate it. but take care, because when we block an activity, we will not block only the activity at the level of the tumor, but at the level of the whole in the whole patient, which could have a lot of secondary effect. So the purpose will be really to try to understand what are the mechanisms allowing the remodeling of the fibers, because the remodeling occurs only at the level of the tumor, and in that case be able to define some therapeutics that will play only on the remodeling of the fibers and only at the level of the tumor. That will be the best.
Okay, fantastic. Thank you. One more question that we've got come in. Can the ultramicroscope light sheet system be used to image larger organs beyond the size of the pancreas?
So this for sure, this ultramicroscope is really amazing, and it allows to image. In fact, the cube that we could use is really large, so enough larger to image a whole mouse for sure, and even larger than for one mouse. So we could clearly use it could be really used to image larger tissue than the pancreas. This for sure. And the only limits, I will say, will be the time, because it takes time. More the tissue is larger is the tissue, more time you need to image. And also, when, like us, you use some antibody staining, it's quite expensive. The amount of antibodies that you need to use is depending on the size of the tissue that you will that we work with, and also the timing, because more the tissue is larger is the tissue, more time you will need to let the antibodies and also to perform the clearing and so on. But for sure, the ultramicroscope will be not limited. It's really an amazing material to work with.
Okay, thank you very much for those really insightful answers to our questions. Thank you again for your time, and thank you again, Sophie, for that really fascinating talk.